Good day. My name is Professor Dr. Bankole Johnson, and I'm the Chief Medical Officer of Adial Pharmaceuticals. I'm delighted to give this presentation on Adial 4301 for the precision medicine treatment of alcohol use disorder in individuals with selected serotonin genotypes. One of the important questions, as you may have gathered, since this is on 5-HT3 receptors, is why are 5-HT3 receptors so interesting and important? Well, let's go right back to the beginning. Let's go back to the beginning of time. At the dawn of time, there were 5-HT3 receptors in the primordial soup. Now, what does this mean? It means that the 5-HT3 receptor is highly adapted. It is also homologous with a large number of other receptors in the superfamily, and also that there is the potential for biologic differentiation within the 5-HT3 receptor itself. So this next slide gives you an idea of basically how far back in time we have seen 5-HT3 receptors. And at the current time, almost every single animal cell contains 5-HT3 receptors. Alcohol exerts its effects through cis-loop ligand-gated ion channels, and this consists of a superfamily of a large number of receptors. Of these receptors is the 5-HT3 receptors I've mentioned before, but we won't be talking about that today because that's not part of the topic. But importantly, these cis-loop ligand-gated ion channels mediate the effects of alcohol by increasing the effects of alcohol to promote dopamine function in the corticomesolimbic system. The next slide shows the complex interaction in the brain between the corticomesolimbic dopamine system and a large number of other modulatory neurons. But most importantly, the 5-HT3 receptor modulates the activity of the dopamine neurons. Now, you'll recall I mentioned that we were going to talk about biological differentiation. Now, in order to talk about biological differentiation, we have to talk about heritability. Alcohol use disorder is a moderately inheritable disorder. Now, you can see on the chart that there are other neuropsychiatric disorders that are more heritable than alcohol use disorder, including bipolar disorder, but alcohol is significantly heritable. This means that alcohol use disorder, in the way we defined it through DSM-5, which is one of our diagnostic criteria, is a heterogeneous disorder. That means that there are various subtypes of individual with alcohol use disorder. The basis of what we're talking about today, which is a precision medicine approach, is really to understand giving the right medicine at the right time to the right patient to have the largest effect in terms of efficacy and the minimization of side effects. So now let's review the foundational evidence that allows us to understand the effects of ondansetron amongst individuals with selected genotypes in terms of response. So the two endophenotypes we're going to discuss, one is drinking characteristics and the other one is genotype. The first study that I want to talk about is a study by Ed Sellers, in which he showed that individuals who drank less than 10 drinks per drinking day formed a particular subgroup of alcoholic, and those individuals were responsive to ondansetron at a dose very similar to the ones which we're using in our current study. The second set of studies are genetic studies, and these have taken place over the last 2 decades. An important genetic study showed that individuals with the L form of the polymorphism at the serotonin transporter were more likely to crave alcohol than those who had the short form. Also, individuals with the L form also varied in terms of platelet uptake. Thirdly, we were able to show that individuals with the L form were more responsive to ondansetron in terms of reducing their drinking behavior compared to the S form. We also have some studies using mRNA which showed that there is a predictable relationship between ondansetron response and some mRNA expression in the way we have actually measured it. So in sum, there are about 6 studies that form the foundational evidence for us to embark on our present clinical trial. What I'm going to do is to show you the phase 2 results of using the L and the S as a differentiator and the LL and TT forms as a differentiator of ondansetron treatment response. The dose of ondansetron here was 4 micrograms per kilogram, and the study population was 283 alcohol-dependent individuals. And here are the results on the Y versus the X-axis, and I'm not going to spend a lot of time, but if you look at this slide, you can see that there is a very strong response for individuals who have the LL and the LLTT polymorphism in terms of their reduction of alcohol-related behaviors. And this doesn't matter how we measured it, whether we measured heavy drinking, whether we measured daily drinking, or we measured some kind of abstinence. As a secondary measure, we also looked at variations at the 5-HT3 receptor. And this is because ondansetron itself binds to the 5-HT3A receptor. So we were expecting that at 5-HT3A receptor subtypes, we would get a more powerful effect of precision medicine using ondansetron. And here we do. And if you look at this slide, this looks at various drinking characteristics. And you can see that the response is even greater at the receptor subtypes than it is at looking at the serotonin transporter. So in our view, this proves a hypothesis that there is a relationship between differentiation in polymorphic variation in ondansetron treatment response amongst individuals with selected genotypes. The next slide is to underscore the point we have made, which is a GMDR analysis. And you can see on the column that there is a consistent response across different drinking types and ondansetron response by genotype. And this was very impressive indeed and allowed us to build up a 5 genotype marker panel. Now, the 5 genotype marker panel is a mixture of genotypes that we found to be responsive in the first study at the serotonin transporter, and also 5-HT A and B differentiated genotypes in that study as well. So this 5 marker study was very important to use in our current phase 3 study. So what are the 3 lessons we learned from the previous studies that are very important? First, an endophenotype of drinking is very important. This is not total alcohol consumption. This is a drinking characteristic that is a type of binge drinking. So individuals who are heavy binge drinkers have this characteristic. The second is that polymorphisms at the serotonin transporter and at the receptor are very important for differentiating ondansetron response. And third, which I only mentioned briefly, the dose that seems to work best is about 4 micrograms per kilogram. This is very similar to the studies that have been successful in the past. And indeed, when individuals have tried to use much higher doses, they have found that individuals taking ondansetron tend to drink more. Now we're going to proceed to the clinical trial itself that we're all here to hear about. So this is a multinational study. It was a 24-week study in which we looked at individuals taking ondansetron 0.33 milligrams twice a day amongst alcohol use disordered individuals with selected genotypes. And this was the 5 genotype panel. The study was 24 weeks long. And one of the things we also did, because we remembered from the earlier lesson, was to dichotomize the endophenotypes by drinking characteristic. So we had heavy drinkers, and we also, and these were individuals who drank less than 10 drinks per drinking day, very similar to the Ed Seller study, and individuals who drank greater than 10 drinks per drinking day, which formed a different subtype. This study was mostly conducted in Europe, and it was a phase 3 study. And the map you can see shows all the places where we actually conducted the study. which was mostly Northern Europe. The inclusion and exclusion criteria were very standard. In fact, they were fairly broad. In total, we had 1,334 subjects enter the clinical trial. Of those, about a third of them were randomized, and there were 303 subjects. 156 subjects were in the ADO4 group, and 147 subjects were in the placebo group. All individuals who entered the study were analyzed in terms of a full analytic system. Now, what were the key entry criteria? You had to have a DSM-5 diagnosis of alcohol use dependence, and you have to have greater than 6 heavy drinking days per month. And we also included individuals who had medium to high risk on the WHO alcohol risk scale. You also had to be a potential responder by being a part of these selective genotypes. And you couldn't be on any antipsychotics or antidepressants, and you couldn't have an unstable type of other neuropsychiatric or substance use disorder. Because ondansetron has been shown to extend the QT interval, we also did not include individuals with heart disease or individuals who had an unstable EKG. I will mention, though, that individuals with QT prolongation to ondansetron have only been recorded in those who took intravenous or intramuscular and not oral, and at doses much, much higher than the ones we're seeing here. In the next slide, you can see the trial design. Basically, all incoming patients received a genetic screen, and once they fulfilled the criteria of being a potential responder, they were dichotomized into the ADO4 and the placebo groups. In this slide, in the trial design, what I'd like to emphasize here is that individuals were in the study for about 8 months. They received screening, which took about 28 days. They were then in the trial for about 24 weeks, and then they were followed up for 4 weeks, about 8 months in total. Individuals during this trial received ondansetron 0.33 milligrams twice a day, but importantly, they also received brief behavioral compliance enhancement treatment weekly, and this was supervised. So all individuals who were administering BBSET or brief behavioral compliance enhancement treatment were trained to do it, and the performance was supervised. This allowed us to provide a stable platform in terms of psychosocial treatment against which we could measure the additional effects of medication. The analysis was done in a very robust and rigorous way statistically. We used a mixed model approach, and the mixed model approach allows us to accommodate individuals who have missing data at random. We also dichotomized the individuals into heavy and very heavy drinkers, and this was part of an important subdivision that was decided a priori, and individuals were balanced into those groups. The secondary variables, or if you like, the additional variables, was again to study what happened in month 6 and to compare that with the other drinking characteristics that we saw. Now, the next slide gives more detail as to how we handle missing data. And I'm not going to state very much about that, except to say that we were very conservative and that individuals who had missing data, we basically corrected back to the initial point, or basically the week 1, on which they came in terms of their data and their drinking characteristics. So this is very conservative. Other parts of how we adjusted for missing data are included in this slide. Now, the next slide shows you the flow trial diagram, and about this time most of you are trying to go to sleep because this is a pretty complex diagram. Don't go to sleep, I'm going to simplify it for you. All this slide tells you is that the groups were fairly well balanced. They were fairly well balanced between ondansetron and placebo groups and between heavy drinkers and very heavy drinkers. Most of the patients in the study were male. And also, about 70% of individuals completed the trial whether they were in the ondansetron group or whether they were in the placebo group. The rest of the characteristics are very similar to previous studies that have been done in alcohol use disorder. The next slide provides the baseline characteristics of the subjects in the clinical trial, which are very much like most clinical trials that have been done in alcohol use disorder. Approximately 2/3 of the individuals who took part in the study were men and about 1/3 were women. The average age was about 48, which is very typical. They weighed about 80 kilograms. And below are the genotype frequencies, which actually correspond very well with the natural frequencies of these genotypes in the wild. The next slide shows the baseline alcohol drinking criteria. And you can tell here that the percent of heavy drinking was very high. So about 65.6% of individuals had very high drinking or very high heavy drinking days. And this was the same whether you were in the ondansetron or in the placebo group. In the very heavy drinker group, though, 76.3% were individuals who had heavy drinking versus 75.8% in the placebo group. So this is very consistent indeed, but the individuals in the very heavy group did drink more and they had more heavy drinking, which is part of the cutoff. But one of the interesting things which I can discuss later when we talk about questions is the overlap between the drinking characteristics of these two groups. The other thing to mention is that the genotypes are fairly well balanced between these groups and the risk criteria were different. So if you look across the groups, and I'll just look at the individuals at very high risk, in the ondansetron group, this was approximately between 31 and 20 compared to 24 in the placebo group in the heavy drinkers, and roughly 58 to 55 in the very heavy drinkers. Now, this was not statistically significant, these differences, although it did show that individuals in the ondansetron group were perhaps a little slightly heavier drinking than in the people who were in the placebo group. And below you can see the pattern of drinking. And as I mentioned before, the endophenotype is all about patterns of drinking rather than total alcohol consumption. The next slide provides an overview of the measures collected during the study. And because we are only presenting the top line data here, I'm just simply going to focus on the drinking criteria. But you can see that we collected a large degree of measures, which is consistent with most modern trials in alcohol use disorder. I wish to remind you again of the primary and secondary endpoints in the study. The primary and the additional endpoints in the study comprised of examining the monthly percent of heavy drinking between the ADO4 and the placebo-treated group in months 5 and 6 combined, and also in month 6 alone. So we looked at the terms in the change in the baseline results. We also looked at the caseness of individuals with alcohol use disorder. That is, what's the proportion of individuals who came in with the DSM-5 diagnostic category of AUD, and did they maintain this diagnostic criteria at the end? And the other piece of data I'm going to present to you is data on the shift in the WHO criteria for measuring quality of life. So I'm going to show you now in the PHDD endpoint results. So when you combine, and I'm going to do this fairly systematically, if you combined the month 5 and 6 data and you looked in terms of drinking, we were not able to find an effect for ondansetron over placebo. However, when we looked at our predetermined dichotomization looking at month 6, and for individuals who had less than 10 drinks per drinking day, we had a significant effect of ondansetron compared with placebo. Indeed, when you look in the next slide of individuals who are drinking greater than 10 drinks per drinking day at the start, it almost seemed although it was not significant, that the individuals with the placebo were doing slightly better. So what this means is that the endophenotype in terms of drinking characteristic is very, very important. The next slide will underscore it, which is a bar graph. And the bar graph shows the reduction of heavy drinking days per month. And you can see in the ADO4 group, this is 77%, and the placebo group is 13%. So there's about a 14% difference here between the two groups. These are proportional reductions, which I think are very important. And if you look at the mean reduction in heavy drinking days, most of these individuals started about 18 days a month at baseline, and they ended up at about 3.8 days in terms of the treatment effect of ondansetron, which I think is very good. So you've converted somebody who was drinking very heavily for about half the month to drinking only a few days of the week. The next slide shows the correlational analysis between months 1 and 6 in the heavy drinking group. Now, why is this important? This is important because clinicians often ask the question, how quickly do you see an effect if you give a medicine for the treatment of alcohol use disorder? And what this shows you is that the relationship between the month 1 and the month 6 changes are highly correlated. So individuals who are going to respond early, and they continue to respond through month 6. In terms of the additional endpoint results, I'm going to go over them very quickly because of time. On the right-hand side of the panel, you can see the DSM-5 criteria, and you can see that there is a significant effect at week 24 in terms of individuals who have less than 2 alcohol use disorder symptoms. This is very important because it's telling you that individuals are no longer meeting criteria for alcohol use disorder. So this is a very important criterion indeed. The next slide emphasizes the effect of looking at AUD in terms of the reduction of patients with AUD criteria. And so we looked at this a different way again. And in terms of these patients, we asked ourselves, what are the numbers of patients who are no longer cases? And this resulted in about 1 in 4 patients in the ADO4 trial, which means one of the most important things that I often say is that if you want to get somebody to reduce their alcohol drinking, put them into an alcohol trial. And the alcohol trial itself has a strong effect in modulating drinking. An important question often asked by clinicians is, okay, you got individuals to reduce their drinking, but did they improve in terms of their quality of life? And this actually shows a very interesting effect for the study in terms of individuals who were heavy drinkers. And these individuals showed at least one shift in terms of the DRL level, in terms of quality of life. Now, is this important? Yes, it is, because the odds ratio is almost 3.5. So individuals who are receiving ondansetron, the heavy drinking group, were about 3.5 times more likely to have an improvement in quality of life. So how do we put all of these results together? An important way to put all of these results together is to look at the 3 areas in which I've shown you to be important. I've shown you that individuals with these selected genotypes taking ondansetron at 0.33 milligrams twice a day are more likely to have a significant reduction in heavy drinking if they're in the heavy drinking group. And this is statistically significant at month 6. I've also shown you that individuals in the same group have less symptoms of alcohol use disorder, significantly again at month 6. Third, I have shown you that these individuals also experience a better quality of life at month 6 of treatment. Now, one of the curious things we did is to do a post hoc analysis, which was to see whether within our genotypes we could find, if you like, a golden nugget of individuals who responded the most. Please be mindful that this is a retrospective analysis, and take it for what it's worth. But what it does show you is that there are certain genotypes that seem to have a very, very high effect in terms of the group in total. Therefore, when we do the second phase 3 trial, we'll probably be focusing more on selecting for these additional genotypes. Now, you may be wondering about safety of ondansetron versus placebo. Now, you might be wondering how safe ondansetron is. The answer is ondansetron is extremely safe. In the data for the study, there were 10 serious adverse events. 7 were in the placebo group and 3 were in the active group. None of these were related to treatment. There were 2 deaths and 2 cardiac events. All of these were in the placebo group, not in the drug group. There was very little difference in side effect profile between AD04 and placebo, and that's shown in the adverse event slide that comes up next. In fact, the frequency of adverse events was very low. And so one of the things that I think is very important to say is not only have we demonstrated efficacy for AD04 in heavy drinkers, but it is also a very safe drug indeed. So what are our conclusions? Our conclusion is AD04 is safe and effective in selective genotypes for the treatment of individuals who are heavy drinkers. This is underscored again not only by the change in heavy drinking, but also the improvement in quality of life and the decrease in alcohol use disorder symptoms. The safety is also underscored by the pill-taking rate. The pill-taking rate was over 95%, in fact it was over 98% in some cases between the AD04 and the placebo group. And the attendance rate at the end was fairly high, which is about 72%. So patients were not only tolerating the drug, they were staying in the study for the 6-month period of time. Now, why is a precision medicine approach very important for the treatment of alcohol use disorder? It allows us to work out who may respond versus who might not respond to a particular treatment and to deliver that treatment to those who may respond, so we don't give unneeded or inefficacious treatment to those who are unlikely to respond. The next is that precision medicine is an important new tool. If this drug is approved, you'll be able to go to your doctor, and if you get you're in the right genotype, you'll be able to get the medicine, in this case AD04 at 0.33 milligrams, with a reasonable likelihood that you're going to get better. Finally, a precision medicine approach improves, or I would say reduces, the stigma of the alcohol use disorder disease. This is because patients will see this as being more medical, and therefore they're more likely to seek treatment, much in the same way as individuals with hypertension will more likely seek treatment, or individuals with other chronic disorders like diabetes. I'd like to thank all of you who participated today in listening to the results of this study. I'd like to thank Adial Pharmaceuticals for embarking on this ambitious trial, and I'd also like to thank all the investigators and co-investigators that made this trial possible throughout Europe and indeed throughout the world. Thank you very much.